because he's not been able to be here today due to some institutional affairs in Modena. I would like to tell you about a story which accompanied us in the last, let's say, 6 years, which I hope that will be inspiring for you and give you hope about the possibility to translate basic science to clinical application for regenerative medicine. And in particular, I would like to talk about this pathology, which is a genetic pathology, which is epidermolysis bullosa. Actually, this story starts in 1975, when Professor Green in Harvard found a way to keep in culture keratinocytes, human primary keratinocytes from a skin biopsy. These are the clonogenic culture that arise from a single cell in rhodamine B or stain in Brightfield. These colonies could rise and cover all the dish. And then this is a single layer of epithelium that can be grown in vitro. And after this phase treatment, we can just take out the epithelial layer from the flask. And this is actually the first tissue ever built in vitro. Colonies, we can divide the colonies in three different types after clonal analysis and divided them from their clonal ability. We have three types of clones: holoclone, meroclones, and paraclones. Paraclones is a very differentiated clones, so we can easily distinguish them, but instead holoclone and meroclone, they are very close in the morphology and clonal size, so we cannot distinguish them during cultivation. But actually, they have many biological differences. In particular, holoclones have high level of P63, and they can self-renew in vitro and in vivo. This is the only clones that can self-renew. So it's actually, we demonstrated in 2017 that holoclones are the clones originated by keratinocyte stem cells. The first application of this technique was done by Howard Green in 1984, when he was able to save the life of patients after massive burns. So he produced autologous layer of epithelium of skin, and then he were able to transplant on the skin on the burn patients. Professor De Luca was there in that years, and then when he came back to Italy, he bring these techniques also in Italy. So he started to cure burn patient that suffer for a massive burns, and then he decides also to try with another epithelium in collaboration with Graziella Pellegrini and Paolo Rama. And they tried to produce a corneal epithelium in vitro. Probably you know that the eye is composed by different epithelia. The white one is the conjunctiva, and here we have the cornea. But keratinocyte stem cells that give rise to the cornea are located only in a narrow zone between conjunctiva and limbus, which is called conjunctiva and cornea, and this zone is called limbus. So, in case of chemical burns, if the limbus zone is maintained, is it possible to proceed with a keratoplasty, and the limbal stem cell residing in the eye could cover again and give a long-lasting corneal regeneration. Instead, in case of a massive burn, in case in which limbal stem cells are lost in these eyes, keratoplastic doesn't work. In this case, the idea was to take one piece, a small biopsy from the limbus of the contralateral eye that was not damaged in this case, to take the keratinocyte stem cell, the limbal stem cell in culture, give rise to a corneal epithelium, and put it on a fibrin gel, and this can be used to and transplanted into the damaged area. Actually, I removed the video of the surgeon operation because it's a little bit too cruent for me. But please believe me that the surgeon can take out all this white area, the pannus, and can add this piece, this fibrin gel with autologous cultured corneal cells and stem cells into the eyes. In this way, we obtain a long-lasting corneal regeneration made by autologous stem cells. So these are some eyes before the treatment. You can see that the conjunctiva cover all the eyes. We have vessels, so we have, in this case, there is a complete loss of the visual acuity. After the treatment, this is how beautiful appears the eye after the treatment, after also a long follow-up. You see that the eyes, we can see the iris, and the most important thing, all the patient restore a complete visual acuity. This therapy is a cellular therapy. It has been registered in EMA in 2015 with the name of Holoclar. Now we actually, in laboratory, we deeply know about the biology of keratinocyte stem cells. We know how to produce skin graft. We know how to produce a fibrin gel with the corneal epithelium inside. And then we decide to move forward and to try to implement a gene therapy approach for a devastating disease, skin disease. Epidermolysis bullosa is a very aggressive disease with a very poor quality of life for this patient. Is due to a mutation of the protein that created the epidermal-dermal junction. So in these patients, we have continuous blistering area due to a continuous detachment of the epidermis from the dermis. You can imagine that you cannot even touch these guys, this baby, because you create a trauma on that skin, in their skin, they go to blisters. So it's a very poor quality of life for the patients, for the family, and they have short life expectancy. They mostly die due to carcinoma. Sorry, there are 3 types of EB. The most severe are the junctional one due to the mutation of the beta-3 chain of the laminin 5 protein, and the other very aggressive is the dystrophic one due to the mutation of collagen 7. First patients, then we decide to transplant. Sorry. I'm sorry. It should be a video that goes on, but it's not working. We decided to apply the same technology that we developed for the cornea and for burns to this patient, but we need also to genetically correct the skin. So for this reason, we took the epidermal stem cells from the patient. We genetically correct in vitro by a retroviral vector. And then we generated a skin graft made with autologous corrected skin keratinocyte stem cells. First patient have been done in 2005, actually, paper published in 2006, where we tried to apply this skin graft only in a small area of the patient, but no blister was ever seen in this transplanted area. And also we have more than 40 years follow-up, no blister occurs in this area. Then, unfortunately, we need to stop. Sorry, there is no one slide. We need to stop actually for the arriving of the EMA law that compare all the stem cell-based therapy to medicines. So all these kind of procedures need the GMP clinics application. So they need a GMP procedure. And then we stopped. We built, Professor De Luca decided to build a center for regenerative medicine in Modena with a GMP facility inside. And in 2005, we were ready to start again, and we produced the first, for another patient with the same mutation, we produced the first skin graft with genetically corrected skin epithelial cells made under GMP condition. Soon as we had the information about the follow-up of this procedure, everything was fine, everything works, an email arrived to Professor Michele De Luca, and it was an email from a pediatric center in Bochum. Please do not photo video in this couple of slides. This mail was telling that there was a baby, a young boy, 7 years old. His name is Hassan. skin and corneal graft. And now, these are the last thing that is now open, is a dermatology unit in which AB could be followed, could be treated. There are dermatologists that take care of them, and all of them, and even that one that are included in our clinical trial. This was a dream for Michele up when he started for epidermolysis bullosa, but now this dream has become reality, and we call it AB Hub. So it's a collaboration, it's a network of professionality that works all together to translate basic science to clinical application. The last thing that is developed is this association, is a no-profit association. It's called the Leali di Camilla, which in English is Camilla's wings, because this baby are called butterfly children because they have the skin so fragile as butterfly wings. This association is a no-profit association that take care of the AB family and promote stem cell research, cell and gene therapy for these patients. Now we have a protocol advice open in EMA for the cure of the junctional epidermolysis bullosa due to mutation of gamma beta 3 change of the laminin 5. And actually we started a phase 1-2 clinical trial to cure another form of AB, the recessive one, due to mutation of COL7 protein. Next year, we would like to start also the gene therapy for another form of epidermolysis bullosa, which is due to the mutation of collagen 17. Of course, all this work has not been made by one person, but is a big team that works all together. And of course, everything is headed by Professor De Luca, that is a wonderful mentor for me and helped me in this phase of my career. Thank you very much for your attention.
Thank you so much.
Thank you.
It's great to see the smile of the child after the treatment.
Yeah, that's the best picture ever.
And we'll skip the question because of time limit, so we'll move to the next presentation.
Okay, thank you.
Okay, I'd like to introduce the next speaker, who's Antonio Campos from the University of Granada in Spain, and he's going to speak to us about clinical implementation of cornea and skin artificial tissues generated with fibrin agarose biomaterials.
Good morning. First of all, I would like to thank to the organizers the invitation to come here and give me the opportunity to share our experience, our modest experience in the clinical implantation of a model of cornea and skin generated by tissue engineering in our lab. Since its appearance in 1993, tissue engineering has globally spread in order to improve therapeutical approaches, current therapeutical approaches, involving a revolution in clinical practice. In this paper that we have just published, we can see an extraordinary increase in the manufacturing, of products generated by tissue engineering. These new therapeutic tools were widely demanded by the population, especially for certain diseases without treatment expectations. The situations generated a use almost without rules that led to a phenomenon called medical tourism. In Europe, until 2003, the use of living cells for any type of treatment was managed by the regulations that were used for organ transplantations without the cells or tissue being considered medicines. It was established by several European directives and regulations when cells and tissue must be considered medicines and when they can be used after a clinical trial or under a hospital exemption. In this page of the European Medicines Agency, we can see that tissue engineering products are included in advanced therapies, and they must follow a strict protocol for their clinical application. What are the phases for the generation of artificial tissue as advanced therapies medicines? First, identification of a medical problem. We need to solve a medical problem. Second, artificial tissue design. Third, preclinical research. Fourth, legal authorization. Fifth, elaboration of the advanced therapy medicinal products. And finally, sixth, application of the advanced therapy medicinal product as a clinical trial or as hospital exemption. I am going to describe here these phases in very schematically in our models or artificial cornea and artificial skin, specifically in the artificial cornea in the anterior lamellar cornea. Of course, there are several problems in the pathology of cornea, corneal ulcers, congenital diseases, surgical complications, limbus pathology, and in skin, burns, tumors, plastic and reconstructive surgery. And these problems demand a substitution therapy. In these papers, we describe our models of cornea and skin, the first one in 2006 and the second in the 2011. Let me now to describe very briefly the model used in both cases. First, isolation and cultivation of corneal skin cells. Second, generation of a biocompatible biomaterial. To make the biomaterial, we selected, after many experiments, fibrin and agarose hydrogels instead of type 1 collagen gels, agarose gels, or only human fibrin gels. The third stage was biofabrication. In this process, to generate, for example, artificial skin, fibroblasts were immersed within the fibrin agarose hydrogel and keratinocytes were subcultured on top. The next step was the process of nanostructuration, which consists in plastic compression and dehydration. The change is obvious, as we can see in the images. This improves very much the biomechanical properties of the tissues. The new nanostructured fibrin agarose model is easier to handle and easier to suture and makes, in the case of the cornea, make the cornea more transparent. This is very useful for surgeons. Of course, after the identification of the problem and the tissue design, the next step is preclinical research, ex vivo evaluation and ex vivo evaluation and in vivo evaluation. For controlling the ex vivo quality, we carried out a cell viability examination, a histological evaluation, and also we carried out a genetic analysis at different levels and a rheological analysis. In these papers, we describe the use of these methods in these tissues. we study cell viability with vital and supravital dyes, and also with X-ray microanalysis in electron microscopy. With this technique, we determine the ionic content in the cells we are going to use to generate the tissues. We can determine very clearly with this method if the cells are alive or if the population of cells are in apoptosis or not. And this is a very important, is relevant for the success in the clinical implementation. Of course, we did also a histological evaluation with light microscopy, transmission, electron microscopy, a scanning electron microscopy. We can see there the layers in the epidermis, the characteristics of the fibroblasts, the elements with electron microscopy, desmosomes, basal lamina, etc., in the new artificial tissues. Of course, we also identified histochemically several molecules present also in these tissues, as plakoglobin, desmoglein, desmoplakin, etc. And we study also genetic characteristics of the cells, gene expression analysis, and of course, protein expressions analysis, what kind of proteins are being expressed, such as different types of cytokeratins, desmoplakin, vimentin, etc. We did also a rheological analysis, and we determined elastic modules, viscous modules, transmittance, absorbance, just to identify very well and to describe very well the new tissue we are considering, we are generating. In vivo experiments, we can observe here, the evolution of the implants in the rabbit. After 6 months, the cornea was very well integrated, as we can see also when using a slit lamp and optical coherence tomography. In the skin, after 40 days, also the artificial tissue was very well integrated. Here we have a histological comparison ex vivo and in vivo, ex vivo and in vivo. And here is ex vivo, in this row, in vivo. And we can see that in vivo, how the epidermis and dermis are very well developed, much better developed. Also, we compare other molecules, ex vivo and in vivo, involucrin, cytokeratins, filaggrin, collagen, all the molecules that were useful to get information about this situation. With all this data, we presented this information to the Spanish Medicines Agency that, of course, obviously follow the European rules in this area. This was possible because in our region, in Andalusia, the regional government has an agency, Andalusian Network for Design and Translation of Advanced Therapies, dedicated to promote the use of advanced therapies. This agency put in contact researchers, clinicians, hospital, university, and prepare all the documents we need to present in the Spanish Medicines Agency. Obviously, that helped very much in the process of the clinical implementation. The medical products, the new advanced therapy, was finally authorized for use by clinical trial in the case of cornea, by hospital accession for compassionate use in scheme. Now, the fifth phase, after legal authorization, is the elaboration of the medical product of advanced therapies in a GMP room previously approved by the Spanish Medicines Agency, according again with the European rules. The laboratory protocol to generate the new model of artificial cornea or skin was adapted to fulfill all regulatory requirements for advanced therapies, medicinal products, manufacturing, according to GMP standards. This is the official page of the clinical trial. Here you can see the medicine is allogenic tissue engineering, nanostructured artificial human cornea in patients with corneal trophic ulcers in advanced stage refractory to conventional ophthalmic treatment. In these papers, the protocol and the characteristic of the clinical trial are described. In the clinical trial, before you can see here the images before surgery and after surgery, and we can